Today's topic is Adrenoceptor Agonists and sympathomimetic drugs. And uh, s- mm, sympathetic nervous system is regulating many organs, and including the blood vessels that regulate the uh, blood pressure. And that is why the adrenomimetics influence the many uh, vital signs, and sympathetic nervous system acts on organs through the releasing of norepinephrine from the sympathetic nerve terminals, and adrenaline released by the adrenal medulla will act uh, through the transfer by the blood. Both of these are uh, influencing the many organs, and uh, the uh, drugs that mimic the sympathetic nervous system are called adrenomimetics. And adrenomimetics or sympathomimetics have the same action as norepinephrine and epinephrine, and they are called sympathomimetic drugs. They can be grouped by the mode of action and by the spectrum of receptors they activate. Some of the sympathomimetics are direct agonists and they activate receptors. Others are indirect because their action dependent on their ability to change the action of endogenous catecholamines. These indirect agents may have either of two different mechanisms. They may displace stored catecholamines from the adrenergic nerve endings, or they may decrease the occurrence of released epinephrine either by inhibiting reuptake of catecholamines uh, or preventing the enzymatic metabolism of norepinephrine. And some drugs have both direct and indirect actions. Both sympathomimetics, direct and indirect, cause activation of adrenal receptors, and the pharmacologic effect of direct agonists depend on the route of administration, the relative affinity for adrenal receptor subtypes, and relative expression of these receptor subtypes in target tissues. The pharmacologic effect of indirect sympathomimetics uh, depends on the increased sympathetic activity and norepinephrine storage and release. The effects of the catecholamines is regulated by cell surface receptors. Adrenal receptors are typical G protein coupled receptors, and G protein has an extracellular end terminus, and uh, and it transverse membrane 7 times. So 1, 2, 3, 4, 5, 6, 7, and that is why it's transmembrane domain, uh, forming 3 extracellular and 3 intracellular loops. 1, 2, 3, uh, and 1, 2, 3. So and it also has an intracellular C terminal. G protein coupled receptors are coupled by G proteins to the various effectors proteins which activate by those receptors. Each G protein is a heterostrimer consisting of alpha, beta, and gamma subunits. G proteins are classified on the basis of the distinctive alpha subunits. G proteins of particular importance for adrenal receptor function include G-stimulating protein of adenylyl cyclase. So G inhibition and G proteins of adenylyl cyclase, and G Q and 11 the proteins coupling alpha receptors to phospholipase C, like we see here. It's GQ. The activation of G protein coupled receptors by catecholamines promotes the dissociation of guanosine monophosphate and from the alpha subunit of the G protein. So actually guanosine diphosphate here, and guanosine triphosphate and then binds to the G protein, and alpha subunit dissociates from the beta and gamma. So this step is shown here, and the activated GTP bound alpha subunit then regulates the activity of its effector. Effectors of adrenoceptor activated alpha subunits include adenylyl cyclase, cyclic GMP, phosphodiesterase, phospholipase C, and ion channels. The alpha subunit is inactivated by hydrolysis of the bound GTP to GTP phosphate, and the subsequent reassociation of the alpha subunit with the beta and gamma subunit. The beta-gamma subunits have additional independent effects acting on a variety of effectors such as ion channels and enzymes. Adrenal receptors were initially characterized pharmacologically, with alpha receptors having the comparative potencies like epinephrine, norepinephrine, and isoproterenol, and for beta receptors having the comparative potencies isoproterenol, epinephrine, and norepinephrine. The development of selective antagonists revealed the presence of subtypes of these receptors, which were finally characterized by the DNA molecular cloning. We now know that the unique genes can code the receptor subtypes this, and the endogenous catecholamine dopamine produces a variety of biological effects that are mediated by interaction with specific dopamine receptors. The receptors are distinct from the alpha and beta receptors and are particularly important in the brain and in the splanchnic and renal vasculature. Molecular cloning has identified several distinct genes encoding 5 receptor subtypes: 2 D-like receptors, D1 and D5, and 3 D2-like, D2, 3, and 4. Further complexity occurs because of the presence of introns within the coding region of D2-like receptor genes, which allows for alternative splicing of the exons in this mediated subtype. There is extensive polymorphic variation in the D4 human receptor gene. These subtypes may have importance for understanding the efficacy and adverse effects of novel antipsychotic drugs. Alpha receptors are coupled via G proteins in the GQ family to phospholipase C. This enzyme hydrolyzes phosphonesides, leading to the formation of inositol 1,4,5,3-phosphate and deacylglycerol. Inositol 3-phosphate promotes the release of sequestrated calcium from intracellular stores, which increases the cytoplasmic concentration of the free calcium. That activates different calci-dependent protein kinases. Activation of these receptors may also increase influx of calcium across the cell membrane. Inositol 3-phosphate is sequentially dephosphorylated, and that leads to the formation of free inositol. Deacylglycerol activates protein kinase C, and which activity modulates the many signaling pathways. In addition, alpha-1 receptors activate signal transduction pathways that were originally described for peptide growth factor receptors that activate tyrosine kinase. For example, alpha-1 receptors have been found to activate mitogen-activated kinase, MAP kinases, and polyphosphoinositol 3 kinase. These pathways may have importance for the alpha-1 receptor-mediated stimulation of cell growth and proliferation through the regulation of gene expression. Alpha-2 receptors inhibit adenylyl cyclase activity and cause intracellular cyclic adenosine monophosphate levels to decrease. Alpha-2 receptors mediated inhibition of adenylyl cyclase activity is transduced by the inhibitory regulatory proteins or G inhibitor. It is likely that not only alpha but also the beta-gamma subunits of G inhibition contribute to the inhibition of adenylyl cyclase. Alpha-2 receptors use other signaling pathways, including regulation of ion channel activities and the activities of important enzymes involving in signal transduction. Some of the effects of alpha adrenoreceptors are independent of their ability to inhibit the adrenergic cyclase. For example, alpha-2 receptors agonists cause platelet aggregation and a decrease in platelet cyclic adenosine monophosphate levels, but it is not clear whether aggregation is the result of the decrease in CAMP or other mechanisms involving GE-regulated effectors. Activation of beta receptors B1, 2, and 3 results in stimulation of adenylyl cyclase and increased conversion of adenosine triphosphate to cyclic adenosine monophosphate, and that increases its concentration. Activation of the cyclase enzyme is mediated by the stimulatory coupling protein GS. Cyclic adenosine monophosphate is the major second messenger of the beta receptor activation. For example, in the liver of many species, beta receptor activated cyclic adenosine monophosphate synthesis leads to a cascade of events culminating in the activation of glycogen phosphorylase. In the heart, beta receptor activated CMF synthesis increases the influx of calcium across the cell membrane and its sequestration inside the cell. Beta receptor activation also promotes the relaxation of smooth muscle, although the mechanism of the smooth muscle effect is uncertain. It may involve the phosphorylation of myosin light chain kinase to an inactive form. Beta adrenoreceptors may activate voltage-sensitive calcium channels in the heart via GES-mediated enhancement independently of changes in cyclic adenosine monophosphate concentration. Under certain circumstances, beta-2 receptors may couple to GQ proteins. These receptors have been demonstrated to activate additional kinases such as MAP kinases by forming multi-subunit complexes within cells which contain multiple signaling molecules. The beta-3 adrenoreceptor is a lower affinity receptor compared with beta-1 and 3 receptors, but is more resistant to desensitization. It is found in several tissues, but its physiological and pathologic role in humans is not clear. Selective agonists and antagonists have been developed but are not clinically available. Dopamine receptors are typically associated with the stimulation of adenylyl cyclase or inhibition of the adenylyl cyclase depending on the type D1, type D2. Like type D1 is 1 and 5, type D2 is 2, 3, and 4. So D1 receptor induced smooth muscle relaxation is presumably due to cyclic AMP accumulation. The dopamine is a vasodilator, and D2 receptors have been found to inhibit adenylyl cyclase activity, open potassium channels, and decrease calcium influx. Many clinically available adrenergic agonists have selectivity for the major alpha and beta receptor subtypes, but not for the subtypes of these major groups. Examples of clinically useful sympathomimetic agonists that are relatively selective for alpha 1, 2, and beta receptors subgroups are compared in this table. And selectivity means that a drug may preferentially bind to one group of receptors at concentrations too low to interact extensively with another group. However, selectivity is not usually absolute, and at high concentrations a drug may also interact with the related classes of receptors. The effects of a given drug may depend not only on its selectivity to adrenoceptor drug, but also to the relative expression of these receptors in the given tissue. Receptors mediated by adrenoceptors are not fixed and static. The number of and function of adrenoceptors on the cell surface and their responses may be regulated by catecholamines themselves, as well as with hormones and drugs, age and number of disease states. These changes may modify the magnitude of a tissue's physiological response to catecholamines and can be important clinically during the course of treatment. One of the best studied examples of receptor regulation is the desensitization of adrenoceptors that may occur after exposure to catecholamine and other sympathomimetic drugs. After a cell or tissue has been exposed for a period of time to an agonist, that tissue often becomes less responsive for further stimulation by that agent. And the other terms as tolerance, refractoriness, and tachyphylaxis have also been used to denote desensitization. This process has potential clinical significance because it may limit the therapeutic response to sympathomimetic agents. Many mechanisms have been found to contribute to desensitization. Some mechanisms function relatively slowly over the course of hours or days, and this typically involves transcriptional or translational changes in the receptor protein level, or its migrational to the cell surface. Other mechanisms of desensitization occur quickly within minutes. Rapid modulation of receptor function in desensitized cells may involve critical covalent modification of the receptor, especially by phosphorylation on specific amino acid residues, association of these receptors with other proteins, or changes in their subcellular location. There are two major categories of desensitization of responses mediated by G protein-coupled receptors. Homologous desensitization refers to loss of responses exclusively of receptors that have been exposed to repeated or sustained activation by an agonist. Heterologous desensitization refers to the process by which desensitization of one receptor by its agonist also results in desensitization of another receptor that has not been directly activated by the agonist in question. Major mechanism of desensitization that occurs rapidly involves phosphorylation of receptors by membranes of G protein-coupled receptor kinase family, of which there are 7 members. Specific adrenoceptors become substrates for those kinases only when they are bound to an agonist. This mechanism is an example of homologous desensitization because it specifically involves only agonist-occupied receptors. Phosphorylation of these receptors enhances their affinity for aristines, a family of 4 widely exposed proteins. Upon binding of aristine, The capacity of the receptor to activate G proteins is mounted, presumably as a result of steric hindrance aristine that interacts with cladribine and cladribine adapter AP2, leading to endocytosis of the receptor. In addition to blunting responses acquired in the presence of the receptor on the cell surface, these regulatory processes may also contribute to novel mechanisms of receptor signaling via intracellular pathways. Receptor desensitization may also be mediated by circuit messenger feedback. For example, adrenoceptors stimulate CAMP accumulation, which leads to activation of protein kinase A. Protein kinase A can phosphorylate residues on beta receptors, resulting in inhibition of receptor function. For the beta-2 receptor, phosphorylation occurs on serine residues, both in the third cytoplasmic loop and in the carboxyl terminal tail of the receptor. Similarly, activation of protein kinase C by CQ-coupled receptors may lead to phosphorylation of this class of G protein-coupled receptors. This circuit messenger feedback mechanism has been termed heterologous desensitization because activated protein kinase A or protein kinase C may phosphorylate any structurally similar receptors with the appropriate consensus size for phosphorylation by these enzymes. The phenylethylamine is the parent compound from which the sympathomimetic drugs are derived. This compound consists of a benzene ring with the ethylamine side chain. Substitutions may be made on the benzene ring, the terminal amino group, and the alpha and beta carbons of the ethylamino chain. Substitution by hydroxy groups at the 3 and 4 positions yields sympathomimetic drugs collectively known as catecholamines. So this is a catechol molecule for the reference. The effects of modification of phenylethylamine are to change the affinity of the drug for alpha and beta receptors, spanning the range from almost pure alpha activity for methoxamine to almost pure beta activity for isoproterenol, as well as to influence the intrinsic ability to activate the receptors. In addition to determining relative affinity to receptor subtype, chemical structure also determines the pharmacokinetic properties and bioavailability of these molecules. Maximal alpha and beta activity is found with catecholamines, drugs that having the 3 and 4 hydroxy groups on the benzene ring. The absence of one or the other of these groups, particularly the C3 position without other substitutions on the ring, may dramatically reduce the potency of the drug. For example, phenylephrine is much less potent than the epinephrine. And and also alpha affinity is decreased uh, about 100-fold, and beta activity is almost negligible except at very high concentrations. On the other hand, catecholamines are subject to inactivation by catechol or methyltransferase, and because this enzyme is found in the gut and liver, catecholamines are not active orally. Absence of one or both hydroxy groups on the phenyl ring increases the bioavailability after oral administration and prolongs the duration of action. And the absence of ring hydroxy groups tends to increase the distribution of the molecule to the central nervous system. For example, ephedrine uh, and amphetamine that are very similar by the structure, and that is why ephedrine is a scheduled drug as well as amphetamine. They are orally active and have a prolonged duration of action and produce central nervous system effects not typically observed with other catecholamines. Increasing the size of alkyl substitutes on the amino group tends to increase beta receptor activity. For example, methyl substitution on norepinephrine yielding epinephrine enhances activity at beta-2 receptors. Beta activity is further enhanced with isopropyl substitution of the amino group in isoproterenol. Beta selective agonists generally require a large amino substituent group. The larger the substituent on the amino group, the lower the activity at alpha receptors. For example, isoproterenol is weak at alpha receptors, so it's purely beta agonist. Substitutions at the alpha carbon block oxidation by amine oxidase and prolong the action of such drugs, particularly the non-catecholamines. Ephedrine and catechol and amphetamine are examples of alpha substituted compounds. Alpha methyl compounds are also called phenyl isopropylamines. In addition to the resistance to oxidation by monoamine oxidase, some phenyl isopropylamines have an enhanced ability to displace catecholamines from storage sites in noradrenergic nerves. Therefore, a portion of the activity is dependent on the presence of normal norepinephrine sites in the body. They are indirectly acting sympathomimetics. Substitution on the beta carbon are typically present in the direct acting agonist, although dopamine does not have a beta substitution. In addition to facilitating activation of adrenoreceptors, this hydroxyl group may be important for storage of sympathomimetic amines in neurovesicles. So these most sympathomimetic have beta hydroxyl group. So the sympathomimetics have prominent cardiovascular effects because of widespread distribution of alpha and beta receptors in the heart mostly and vessels. Uh, Also in the neural and hormonal systems involved in blood pressure regulation. The net effect of a given sympathomimetic in the intact organism depends not only on its relative selectivity for alpha or beta receptors and its pharmacologic action at those receptors. Any effect these agents have on blood pressure is counteracted by compensatory barrier reflex mechanisms aimed to restore homeostasis. The effect of sympathomimetic drugs on blood pressure can be explained on the basis of their effects on heart rate, myocardial function, peripheral vascular resistance, and venous return. The endogenous catecholamines, norepinephrine and epinephrine, have complex cardiovascular effects because they activate both alpha and beta receptors. It's easier to understand these actions by first describing the cardiovascular effect of sympathomimetics that are selective for a given adrenoceptor. So alpha-1 receptors are widely expressed in vascular beds and their activation leads to arterial and venous vasoconstriction. Their direct effects on cardiac function is of relatively less importance. A relatively pure alpha agonist such as phenylephrine increases peripheral arterial resistance and decreases venous capacitance. The enhanced arterial resistance usually leads to a dose-dependent rise in blood pressure. In the presence of normal cardiovascular reflexes, the rise in blood pressure elicits a baroreceptor-mediated increase in vagal tone with slowing of the heart rate, which may be quite marked. However, cardiac output may not diminish in proportion to this reduction in heart rate, since increased venous return may increase stroke volume. Therefore, direct alpha adrenoceptor stimulation of the heart may have a modest positive inotropic action. The magnitude of the restraining effect of the barrel reflex is quite dramatic if barrel reflex function is removed by pretreatment with a ganglionic blocker, for example, trimethophan. The pressor effect of phenylephrine is increased approximately tenfold and bradycardia is no longer observed, confirming that the decrease in heart rate associated with the increase in blood pressure induced by phenylephrine was a reflex in nature rather than a direct effect of alpha receptor activation. Patients who have an impairment of autonomic function exhibit this extreme hypersensitivity to most pressor and pressor stimuli, including medications. This is to a large extent due to failure of barrel reflex buffering. Such patients may have exaggerated increases in heart rate or blood pressure when taking sympathomimetics with alpha and beta adrenergic activity. So heart rate will increase in case of the beta and blood pressure increase in case of the alpha adrenergic activity. This, however, can be used as an advantage in their treatment. The alpha agonist midodrine is commonly used to ameliorate orthostatic hypertension of these patients. There are major differences in receptor types predominantly expressed in the various vascular beds. The skin vessels have predominantly alpha receptors and consist in response to epinephrine at norepinephrine, as do the splanchnic vessels. Vessels in skeletal muscle may constrict or dilate uh, depending on where the alpha or beta receptors are activated. The blood vessels of the nasal mucosa express alpha receptors, and local vasoconstriction induced by sympathomimetic explains the decongestant action. So I think that is all for today, and thank you for your attention, and see you next week.